Welcome, life science learners, to an installment of Life Sciences where we're looking at scientific experiments and scientific methodology. I'm sure you guys have spent some time at school trying to figure out the reasons for why things happen or trying to solve a problem. And often we end up looking at a process called scientific method. So over the next few episodes, we're going to look at the principles behind how we set up an investigation, how we collect data, how we interpret that data and present that data in a report where it's easier for us to be able to draw a conclusion. So I hope you guys settle in, let's engage with this, and let's see how we go about solving scientific problems. Okay, so we're going to look at designing a scientific investigation today. So, have you ever thought about the following? Why do plants grow upwards? Or, for that matter, why do leaves have a green color? And often this is a question that we asked, and we've got to kind of figure out, well, what is the explanation for that? And so for someone to be able to prove the reasons for why a plant grows up or why leaves are green, we've got to do a specific process called research, and we've got to carry out an investigation to actually determine the factors or the reasons why a plant has green leaves or why it chooses to grow upwards. And hence, let's look at that process today and try and understand the method behind trying to solve a question or a problem. So, for you, I've got a challenge question today, and we're going to look at this at some point at the end of this session. So the question is, what is the first step in a scientific method? The options are A, to ask a question, B, a conclusion, C, to formulate a hypothesis, or D, to make an observation. So I'm going to leave that with you guys to kind of simmer over as we get through this lesson. Let's look at the concept behind scientific method. And in this illustration, I have essentially laid out the entire process of what goes into scientific method. So at the start of any procedure, the first thing we need to look at is, what is the question that we need to ask, or what's the question that's being asked? And in this case, I've asked you, okay, have you ever thought about why do plants grow upwards? And that's a question that I've asked. And often you end up getting questions posted to you in terms of why do things happen? So that's a question that you don't have answers to. So the next step would be that you go and read about that. So you do your research. And research could be reading, and you go into Google and you go and type in your question, and you look at other resources that are available. And sometimes there's often research done on a similar topic, or someone else that's currently working on something that's relevant. So you start understanding other relevant information that's a link to your question. And this does help in you being more focused in your research. So let's read. Let's try and understand what other research is happening around your specific area of focus. Then you've got to construct a hypothesis. And essentially, we've got to look at what a hypothesis is. And essentially, a hypothesis is probably I would use a guess, a prediction, probably what do you think would happen? So you make a statement, and that statement needs to be tested through carrying out an investigation. So in the case of the question that we asked at the beginning, why do plants grow upwards? Well, we've got to come up with a hypothesis. And that hypothesis is plants grow upwards because they require light, or plants grow upwards because they don't require light. So we've got to now establish the reasons behind that. So now we've formulated a question, and we've got to now test a series of experiments that need to be conducted so that we can get an answer to that. So let's look at the next process. So we've got to test that with an experiment. So we've got to design an experiment where we can test and follow a specific method. So that would require a method where we carry out an investigation to determine whether light is needed for plants to grow upwards. The next step would be, let's see if that method actually produces results And has that procedure worked? So we conduct an investigation using a scientific method, and then we look at whether that has worked. And there's two possibilities. Yes, it has, or no, it hasn't. If it has, we've got to analyze the data. And we've got to use the data that we've generated from that experiment to draw a conclusion. Once that is done, we can either align our results with our hypothesis, meaning that the results now support the specific hypothesis that we initially made, or it does not, and we've got to review our hypothesis. So essentially, we could, at the end, after collecting the information, realize that yes, the information supports the hypothesis, or no, it does not. And hence we've got to now evaluate our initial statement around the hypothesis. So that takes on then to the next process of scientific method, is communicating your results. And you'll often see that when you're doing research, you have to present a report. And that report is essentially a method of sharing your information to other people that will read that and have a better understanding of what you did, or for them to be able to compare their results or their findings to yours to see if there is some similarities or there's differences, so that there's more collaboration in science. And currently, if you look at globally, there's lots of research that's happening. For example, on trying to establish a vaccine for COVID or a cure for AIDS. So scientists around the world worked, but they work in isolation, but they often collaborate. And that's part of sharing their information. And in science, you will see that that brings information together, that helps scientists to be able to communicate and collaborate, making discoveries much more effective and finding solutions much more easier. So that's the communication bit. However, there is a possibility that when you carried out your investigation, it did not work. So what happens then? So then we look at, we analyze, we reflect, we look at areas that have had problems. So we call them troubleshooting areas. We carefully check that all the steps have been done and that correct processes have been followed. And then we go back to reviewing the test. When we go and look at our hypothesis, and if we've got results now and those are not aligned to the hypothesis, we may need to go back and review the experimental data, the background information. We possibly need to ask more questions. We need to review our hypothesis, and probably, most definitely, I would conduct the experiment again. And that's essentially done to ensure that we produce now results that are more reliable and we have more valid results. Two terms that we're going to refer to later on and try and analyze and understand what these terms mean. Right, so that's the overview. So generally, we need to follow the following steps, and if we take these steps into kind of consideration, we would have some idea as to how we would set up a scientific method. So the first is you've got to make observations, and you've got to set up a question. You've got to do reading around that, and that could be on similar research, it could be on facts or concepts associated with your research. In our example, what do plants need for growth? What do plants require in terms of for them to grow effectively? What is there in light that causes plants to grow? Do plants have hormones? So we've got to ask all these questions that will point us to the reasons possibly why plants grow upwards. Okay, we've then got to formulate a hypothesis, and then that's going to be an educated guess, probably a prediction that you need to establish a set of experiments. So you make a prediction, and that prediction would be either supported or not. We will test the hypothesis, and that's by conducting an investigation. So as I said, you would have to have an experiment set up, so we investigate, and that's through scientific experiments. And once those investigations are carried out, they give us results, and those results should be analyzed and conclusions drawn. As I said, it's important for us to be able to report on these and share your results. Cool. So, what are the key words in this section? And as we get into this next segment, we've got to look at these terms, and so I'll unpack these as we get through. So I've mentioned the term hypothesis, and hypothesis is essentially an educated guess or a prediction around what might happen. And these often would need to have the independent variable, as I've mentioned below here, and the dependent variable. And these two variables would be what we actually need to consider in our experiment. Okay, we've got to have an aim, and that aim is essentially an investigative question around what we're trying to establish. We've got to know what our independent variables are, and that's essentially what we are testing or what we have control over. So what are we testing? That's the independent variable. We also need to identify how do we record results. And so what is our dependent variable in this? So recording, what do you record? And if you are recording the height of the plant or the light intensity, that would be the recordings that talk to the dependent variable. We also need to keep our control variables in mind, and those are what keep the experiment accurate and reliable. At the end, we've got to look at accuracy in terms of what did we do that ensured that our experiments were reliable. So we look at accuracy. We look at the reliability of our results, which essentially points to if you were able to do this experiment again, would you get the same results? So that's reliability. And how valid are these results? And validity talks about, is it valid enough for you to be able to share these, and so that someone else does these experiments and gets the same results? And that essentially talks to what we refer to as the validity of an experiment. So guys, we've looked at some key concepts. We've looked at the overview in scientific methodology. It's been an intensive session where we've been exposed to some new terms, some familiar terms, some not so. So I think you guys deserve a good break. Let's have a short break, refreshen up, and we'll see you in a short while. Welcome back, life science learners. So let's continue with today's installment of scientific methodology. Let's get straight into it and try and understand the process of scientific method. So let's look at what we've got on the slide. So it's important for us to know what is scientific method. So if we were to analyze this and unpack this concept, we would read as follows. The scientific method is defined as a method of research in which a problem is identified, relevant data is gathered, a hypothesis is formulated from this data, and the hypothesis is empirically tested. In simple terms, the scientific method is a way for scientists to study and learn things. So guys, if we were trying to establish what is the chance of us having heads and tails, we would need to come up with some kind of question. And that question is, what is the probability of having a head or a tail on a coin? And in order for us to be able to prove that it is 50%, we can guess that it's 50%, but we would need to test that. And so there is information that you know already that, a coin has two sides. There is a chance of it landing on a head or a tail every time you toss it. How do you present all of that in a research paper? Or how do you write a report that tossing a coin gives you a 50% chance of having a heads or a tail? So we would need to carry out a scientific method. And so we would need to write a question out, we would need to write a hypothesis out, and we would need to write an aim. So if I toss a coin, the question is, what is the chance of me having a head? And probably you're going to say, well, 50% or 1 in 2 chance. Can we test that? And so we would need to design a hypothesis. So my hypothesis is going to be, when I toss a coin, I have a 50% chance of having a head. Or, when I toss a coin, I have a 100% chance of having a head. One of these two is going to be tested either positively correct, or we're going to get a negative result at the end. So we would need to review that hypothesis. So that would be the hypothesis. It has the independent variable, which is the chance of having a heads or tails, and the dependent variable would be the percentage chance. So I said either 50% chance of having a heads or 100% chance. So we're going to measure that. We also need to ensure that we record our results. So that would be the dependent variable. So I will toss this coin a few times and count the number of times I get a head and a tail. And I'll record that as my data. But before I do that, I need to write down an aim. So the aim would be the investigative question. When I toss a coin, what is the probability of me getting a head? So that's a simple question. And so I've got to carry out a method now that's going to generate data. And that's going to be the data that's going to talk to the hypothesis and answer the question around what is the aim of the investigation. So again, a simple concept, and you can apply that concept in most scenarios where you're trying to establish the scientific method to trying and solve a problem or question that you have no answer to. So let's go back to more processes around scientific methodology. So we've got to ask ourselves, what is a research question? And as I said, it's a question that can have answers that will generate a specific focus area. So a research question is an answerable inquiry into a specific concern or issue. It is the initial step in a research project. The initial step means after you have an idea of what you want to study. The research question is the first active step in the research project. So often at school, you're going to be given a research project, and it could be on investigating the effect of pollution in your area. So you've got to understand, what is the focus of that research? So is it in your area? Is it in your street? Is it the littering of paper or plastic? So you've got to be very focused in that, so that your method becomes very defined and you're able to generate data that points specifically to your question. So it's important that you focus on a unique research question. And we often at school will ask our learners to come back to us and present their research question, so that we have an understanding of their thinking and we can point you more specifically to a more pointed direction in your research. So it's often good to ask someone else, what do you think about my research question? Am I clear enough? Is this going to get me kind of information that I want to be able to answer my project or to be able to get information? So ask an intelligent research question, and that must be very pointed. Pointed in that it is clearly testing an aim. Okay. So here we get to the next bit. What is the aim of an investigation? So the aim identifies the purpose of the investigation. It is a straightforward expression of what the researcher is trying to find out from conducting an investigation. So with the example of, have you ever thought about why do plants grow upwards? We've got to write down a specific question that relates to the aim. So the aim would be to investigate why plants grow upwards. And that's very clear. It talks to the hypothesis, it talks to our research question as well. So the aim is essentially the intention of what you need to establish in your experiment. And that's very important, is that your aim should be very clear and relate to the intention of what you need to establish at the end of your investigation. Okay. What is the hypothesis of an investigation? So guys, often we get told that a hypothesis is a guess, it's a prediction. Essentially, a hypothesis is a statement around what you possibly think might happen, or what do you anticipate or predict that might happen. And the components of a hypothesis needs to include, one, the independent variable. It should include dependent variable, and it needs to link to the research question. So a hypothesis is a precise, testable statement of what the researcher predicts will be the outcome of the study. So my prediction is that plants will grow upwards towards the light and increase in height. That's a testable statement. We would need to work around an aim and then a method to test that. So that's a statement that I predict that plants will grow upwards. Okay, so this usually involves the proposing of a possible relationship between the two variables. As I mentioned, the independent variable as well as the dependent variable. So what is the independent variable? This is the variable that the researcher changes. So this is what you have control over, and it's that variable that you're able to manipulate and change. So if I wanted to test why plants grow upwards, I have the ability to be able to control the factors that cause plants to grow upwards. Things like light, water, carbon dioxide. So those factors would be things that I have to manipulate. In this case, the dependent variable is what we're going to use to measure the results, but the independent variable is the variable that we are manipulating that has an impact or an effect on the other variables. So let's look at what that means. The dependent variable is what the researcher measures. And so if you think of measuring the height of a plant, measuring how quickly it grows, those are measurables that talk to results. And they tell us if there's an effect or there's an impact of the manipulating the independent variable. So the dependent variables often is what you measure that helps you to draw a conclusion or to be able to draw an inference from. So we got to know clearly that in our hypothesis we've got to include both the dependent variables and the independent variable. So what are the variables in a scientific investigation? And here we talk to specifically the types of variables. So we talk about the things that are changing in an experiment are called variables. So variables refer to variation. A variable is any factor or trait or condition that can exist in differing amounts or types. An experiment usually has three kinds of variables. As I mentioned, the independent, the dependent, and the control variables. And so we're going to unpack these as we get into the next slide. So the question is, what is the independent variable? Again, I'll explain this once we get through this slide. An independent variable is exactly what it sounds like. Again, a variable that stands alone. The word independent means standing alone. So it isn't changed by the other variables. So it is independent of the other variables and is not affected by the other variables. Okay, you are trying to measure this. So in terms of when you are carrying out an investigation, it's something that you can manipulate. So when I say manipulate, I mean that you physically can control. So you can physically control this and you can test that. The independent variable is the cause factor, meaning that it is the factor that causes the change, or it's the factor that will give you results. So it will lead to the dependent variable changing. So changing the light will cause the plant to grow. Removing the light will cause the plant to not grow or for the height to remain the same. So that's the manipulation of the independent variable. The independent variable is a variable that we can change or control in a scientific experiment, and that's the word I used in terms of change or control, referring to manipulation. It will represent the cause or reason for the outcome. So often in an investigation, we need to establish what is going to be the reason why that plant is going to increase in height, or what are the factors that make the plant leaves green. And so that will be the independent variable. So we've got to look at, is chlorophyll the factor that makes the plant green, or is it the oxygen and carbon dioxide that is used and produced by the plant? So we've got to establish exactly what is that factor that changes or causes the plant to actually become green or grow. And that's the factor that we refer to as the independent factor. Okay. The next variable is the dependent variable. And the word dependent means something that you can count on. Okay? So the dependent variable is one in which we can test the scientific experiment in order to get its values. So essentially, it's what's going to produce the results. The dependent variable is obviously dependent on the independent variable. So essentially, whatever changes because of the independent variable is dependent on that variable. So we refer to those as the dependents. So when a plant increases, gets more light, it's going to increase in height. So when it increases in height, it's because of the light. And so that height increase is dependent on the light, correct? It's the independent variable, which is the light, and the increase in the height is the dependent variable. Okay, so the factor that is being measured after the input variable has been enacted. Example, the temperature of the patient at different time intervals in degrees Celsius. So we talk about measuring the temperature of an individual, and you know that as the temperature increases, you feel much hotter. So the temperature of an individual increasing would be the dependent variable. So if someone had an infection and if you wanted to measure their body temperature because of that infection, measuring the dependent variable would be, let's monitor the body temperature over a period of time, and that would be because of the infection causing the change in the body temperature. So that's one way of us measuring the dependent variable. Okay, we're going to try and wrap this slide up by looking at the control variables. And the control variables are the ones which the scientists keep constant or hold constant during an experiment. Thus, we also know that the control variables are the constant variables, or sometimes referred to as the control only. However, control variables is not part of an experiment, but it doesn't affect the variables, but it is important because it has an effect on the results. Remember that it is not the same thing as the control group. So let's try and understand that in a nutshell. The control variables are things that you control and make the experiment fair. So if you are comparing the effect of light of two plants, you've got to ensure that you keep the amount of water the same, you keep the type of plant the same, you also ensure that the amount of soil that they kept in is the same. So those variables that you keep constant and you subject both the samples to are the control variables. And so that ensures that your experiment produces a fair test. And so as we end on the note of a fair test, I think you guys deserve a break, and then we'll look at what is validity, fair test, and trying to unpack some of the applications of these concepts. So a quick break, and then we'll see you guys at the end of that. Welcome back, life science learners, to our final installment for today. Let's continue looking at scientific methodology. Before we went into the break, we spoke about the independent variables, we spoke about the dependent variables, we ended off looking at control variables. I mentioned that it was important for us to be able to have these variables controlled. Essentially, the control variables are factors that you keep constant so that it ensures that your results are fair and they're reliable. So what does that mean? It means that if we can control our variables and keep them constant, we're able to produce an experiment that has a fair test. So let's look at the concept of what a fair test is. So a fair test is an investigation where one variable, which is the independent variable, is changed and all other conditions, meaning the control variables, are kept the same or constant. What is measured is referred to as the dependent variable. Let's try and unpack that. So we often have an experiment where we're testing what the independent variable is. We also need to ensure that we're able to measure some variable which we refer to as the dependent variable, and that talks to the effect of the independent variable. But to ensure that our experiment is fair and consistent and produces the same results, we need to keep those other variables constant. I also need to differentiate between the term control, test, and control variables. So let's go back to our experiment where we looked at why plants grow tall. And so if we assume that light is the reason that plants grow tall, or if it's hormones that's the reason why plants grow tall, we would need to ensure that we have a control test, which is one set that is not exposed to the independent variable. So we often have to make sure that we remove the independent variable from that setup to see if that variable has any effect on the plant, or in this case on that group. So in a control test or in a control group, we often remove the independent variable or the variable that's going to cause the impact or change on the dependent variable. So in a vaccine trial, you will have a group that receive the vaccine. However, there will be a group that does not receive any vaccine, and that's the control group. And we want to see if that group shows any symptoms or any effects of having not received the vaccine. So we want to make sure that everything else is the same, but we remove the independent variable from that group. And we refer to that group as the control group, and that group establishes what the other variables are going to have an impact on them. So it's important for us to keep the control variables constant, but it's also important for us to be able to have a control group, and that's a group that we're able to compare our test results to look at the effect of the independent variable as compared to the group which does not have the independent variable. And hence it's important for us to be able to have controls and to have a control group. Okay, so a fair test is a test in which the controls are all but one variable when attempting to answer a scientific question. Only changing one variable allows the person conducting the test to know that no other variable has affected the results of the test. And as I mentioned, that this is the best way to know and isolate the impact of the independent variable, is for you to have a control group as well as having the control variables kept constant. Some textbooks, some teachers refer to that as your fixed variables, things that are kept constant. Okay, cool. So those will rely or will talk to what we refer to as the reliability and the validity of an experiment. So we often refer to things as being reliable or that they're valid. And reliable means you can count on. Valid means it's true, it's accurate. So let's look at these terms in the context of scientific methodology. So reliability and validity are both about how well a method measures something. So let's look at reliability. It refers to the consistency of a measure, whether the results can be reproduced under the same conditions. So when we talk of something being reliable, if you talk about an appliance being reliable or a brand name being reliable, we often means that that brand or that product gives you the same results year after year, or it gives you consistency in its performance. So you often hear of people saying, well, it's a very reliable brand or it's a reliable car, and that reliability they refer to as being something that is consistent. It gives you the same value for money on a long-term basis. So in scientific methodology, we need to ensure that our results are reliable. So if I conducted my experiment and I had written down a method, and if I shared those results with another scientist and they've collaborated with me, they are able to conduct the exact same experiment and get the exact same results or similar results. If that's possible, we say that the results are reliable, meaning that it can be replicated to produce the same results. That is what reliability is. So it's a fundamental aspect to having a fair test. What is validity? And if we talk of validity, it basically refers to the accuracy of a measure, whether the results really do represent what they are supposed to measure. So accuracy points to the way in which you produce valid results. So when conducting a scientific method, were you accurate in the quantities that you've measured? Were you accurate in ensuring that the temperature was maintained constant? So what apparatus did you use to ensure that you measured accurately? How did you monitor the process? How did you measure time? So that's accuracy. So accuracy again leads to what we call fair testing and producing reliable and consistent results. So let's look at reliability and validity in a comparison table. So when we look at reliability, we know that in first-hand information and data, have I tested with repetition? If you've tested with repetition, we say that you have now produced reliable results. In secondary information and data, so that means if you're doing research through information and collecting data from different sources, how consistent is that information from other reputable sources? Is the data presented based on repeatable processes? So essentially, if you went and did secondary research, and if you went into the internet and you kind of Googled information around a similar topic, are those experiments producing the same reliable results that you have got? If they do, we say that that information is reliable because we're looking at the reliability of yours and we're linking that to that. And hence it increases the reliability when we talk of secondary information. Let's look at validity in the context of primary or first-hand information and secondary. So does an experiment procedure design actually test the hypothesis that I wanted to? Have all the variables been identified and controlled? And if you answer all these questions correctly, we refer to that as being now a valid experiment producing valid results. We can also look at validity in the context of secondary information. So do the findings relate to the hypothesis or problems? So when you read other information or other published sites, do they relate to your primary information or your experiment? And that is what produces valid results. So how do you know your results are valid? If you compare them to a published journal, and if they produce similar results, we say that this experiment has now produced valid results. Okay. So let's look at the application of this. So Spur Bubbles is about a product that SpongeBob has created. Let's look at this. So Patrick and SpongeBob love to blow bubbles. Don't do we all? Patrick found that some Super Bubble soap at the Sale Mart, the ads claim that the Super Bubble soap will produce bubbles that are twice as big as the bubbles made with the regular bubble soap. Patrick and SpongeBob made 2 samples, okay, of the same solution. One was made with 5 ml of the Super Bubble soap, and the other was made of 5 ml of water. So it was mixed with 5 ml of that and water. The other was made with 5 ml of regular bubble soap and 5 ml of water. So they've created a solution having 5 ml of that and the Super Bubble soap, and another solution having 5 ml of the regular bubble with 5 ml of water. Patrick and SpongeBob used their favorite bubble wands to blow 10 different bubbles and did their best to measure the diameter of the bubbles. So they measured the diameter of the bubbles. So that's talking to what they measured as they popped on the table. So as these bubbles popped in the table, they produced a little a diameter which they measured. The results are shown in the data table on the next slide. So this information was captured. So here we see the results shown in the table, and here's the Super Bubble which they had purchased, and here's the regular soap bubbles, and here we look at the diameter of these bubbles which have been recorded along this, and they've attempted to do this for 10 different bubbles, and we can see clearly from this the information is presented and that has been recorded in the table. So let's look at this and try and interpret how this information talks to the aim of what these guys were trying to establish. So what is the independent variable in this investigation, and what is the dependent variable? So if we go back to that slide, and if we were to look at what is the independent variable, remember that these guys were looking at the sale mart, and they picked up types of bubbles. So they were looking at the regular bubble soap, okay, and they were comparing that to the super bubble soap. So clearly, if we were to look at what our soap, so that's the one that we're talking about, so that's the super bubble and the regular bubble soap. So that's going to be the type of soap or the type of bubble would be the independent type of bubble. We said that's the independent variable because that's what we're testing. Okay. So that's the independent variable. We also know that they've got to measure these, and so here you see that they've measured the diameter of the bubbles, and so that we refer to as our dependent variable. So that's our dependent variable. So if we go back to the table, we also see that in the table. So here we see that these, which is often in the table headings, are your independent variable, and we look at that in the table, and we can pick that up from the table as well. And further down, we see that the measurement of the diameter of that bubble when it landed on a flat surface was the dependent variable. So the diameter is the dependent variable as we see illustrated in this table. Cool. So let's move on to the next question. Look at the results in the table and calculate the average diameter for each bubble solution. So here you've got to go and use a calculator to calculate that. And so it means that it's important for us to be able to use information from the data table to calculate exactly the diameters. And in this case, you guys would need to add up the sum of all these 10, and you would need to divide that sum by 10. And so essentially it's the total of these. So you've got to add them up, and you've got to divide by 10, and you've got to do the same for the data collected for the regular soap, and that would also be divided by 10 to produce what we call the average. And why is the average done? Remember that in science we repeat experiments or we calculate the averages, and those averages often refer to reliability or consistency of your results. So it's important for us to be able to calculate averages when carrying out a scientific investigation. So I'm going to leave that to you guys to calculate. We're going to move on swiftly to the next question. What do you think SpongeBob and Patrick's conclusions should be? So remember that when they looked at this experiment, they were looking at which of the two. So if I go back to the aim of the experiment was to determine if this super bubble soap was going to produce larger bubbles than the regular bubble soap. And so if we go to the data table, and if you look at the results, and if you calculate the averages, you will see that the super bubble soap produces an average that is significantly higher or larger than the average produced by the regular bubble soap. So again, when we go to our conclusions, we've got to be able to use information. So here you've got to calculate that, and so you would have got a value, and that value would be an average for that. So I'm going to give you an average. Let's say it was 5.10 centimeters, where the regular was, I'm just guessing this for now, 4.10. We can see that clearly that the average for the super bubble was significantly higher. Okay. Are these results reliable? Why or why not? And many of you will choose to either go, yes, it's reliable, no, it's not. There are many flaws in this design of the experiment, or it was only done for 10 bubbles. So we're not really sure if this was reliable, but I can look at the method. So they've used 5 mLs of both the samples, which was fair. They've diluted them in 5 mLs each, which was again controlled. So from a fairness and reliability, that was valid. They also measured 10 bubbles. So it wasn't a measure of comparing 2 bubbles or 1 from each. They've looked at an average. Yes, it might not be consistent, it might not be enough, but we can increase the number of bubbles to give you more reliable results. So for me, I think that these results are quite reliable in that they're producing significantly good results that we're able to compare and contrast and draw a conclusion. Okay, so folks, we're getting to the end of the session. As we overview what we've done today, we've looked at scientific method, we've looked at writing a hypothesis, we've looked at how we lay out an experiment, we've looked at variables, most importantly independent, dependent, and your control variables. We've also spent some time looking at what a fair test is and how reliability and validity are important to ensuring that results at the end generate reliable information. So from my side, it's been a good session. I'll see you guys soon where we talk about how to manipulate data in a scientific method. From me over to you guys. Take care. Go well.